Good morning. It's April 28th, and this is your daily brief in science. Here's everything you need to know. Austria is taking a significant step to combat honey fraud. The country's Health and Food Safety Agency, AGES, has launched DNA testing initiatives that align with the European supermarket chain SPAR's decision to test its honey products. The DNA testing, developed by Synsoma, can identify unique DNA profiles in honey, aiding in the detection of fraud. However, experts caution that this method may not catch all forms of adulteration and requires thorough validation. Synsoma has created an affordable DNA test for beekeepers, priced at €94 or about $103, making it a cost-effective alternative to traditional pollen tests. This test also includes a QR code that allows consumers to trace the plant species in their honey. Corinna Wallinger, head of sales at Synsoma, acknowledges the challenges in standardizing detection methods for honey adulteration. She emphasizes the need for continuous innovation in technology to keep pace with the evolving tactics of fraudsters. The standardization of testing methods is crucial but will require time, as counterfeiters are constantly adapting their techniques. In response to the rising issue of honey fraud, Brussels has formed an expert group aimed at standardizing detection methods by 2028, enhancing traceability for producers and importers. Recent investigations revealed a concerning trend. 46% of honey entering the EU from 2021 to 2022 was flagged as potentially adulterated, a sharp increase from just 14% recorded between 2015 and 2017. This highlights the urgent need for effective testing measures. Matthias Kopetsky, a beekeeper in Vienna, has expressed frustration over the challenges posed by lower-priced imported honey, complicating the market for local producers. The EU is currently the second-largest honey importer globally, sourcing primarily from Ukraine, China, and Argentina, while also being the world's second-largest honey producer after China. ImmVira has made headlines with the recent presentation of Phase 1 clinical results for its oncolytic herpes simplex virus product, ImmVRC-5252, which targets malignant glioma. This aggressive form of brain cancer has a 5-year survival rate of less than 5%, highlighting the critical need for new treatment options. As a global biotech company, ImmVira focuses on developing innovative therapeutic solutions using engineered oncolytic viruses and exosomes through its OVPENS platform. ImmVRC-5252 has already received investigational new drug approval in both the US and China, along with orphan drug designation from the FDA, which aids in its clinical development process. The drug is administered intracranially using convection-enhanced delivery, or CED, allowing for even distribution and effectively bypassing the blood-brain barrier. The clinical trial, conducted in collaboration with Duke University, has involved 5 patients with recurrent high-grade glioma, with 3 patients in Stage 1A completing the dose-limiting toxicity period at a dosage of 5 × 10 to the power of 6 pFU. Cytokine analysis of cerebrospinal fluid after infusion has shown dynamic immune responses and biological activity, with no serious adverse events reported. The side effects experienced were mild, primarily grade 1 to 2 adverse events such as fatigue and cognitive disturbances, suggesting a favorable safety profile for ImmVRC-5252. ImmVRC-5252 is specifically engineered to target malignant gliocytes, while also enhancing the immune response through the use of PD-1 antibodies and IL-12. Dr. Grace Guoying Zhou, ImmVira's chairwoman and CEO, has reiterated the company's dedication to addressing the unmet medical needs in malignant glioma through their innovative therapies, leveraging the unique properties of HSV1 in collaboration with Duke University. Nanotechnology is making waves in cancer treatment by allowing for precise targeting of cancer cells, significantly reducing side effects compared to traditional therapies. At the forefront of this innovation is the University of Cyprus, which has developed a patented nano-vehicle specifically aimed at treating triple-negative breast cancer, a particularly challenging type of cancer. These engineered nano-vehicles are designed to deliver drugs directly to cancer cells while sparing healthy tissues, thereby enhancing treatment efficiency. The success of lipid nanoparticles during the COVID-19 pandemic for mRNA vaccines has highlighted the potential applications of nanotechnology in cancer therapeutics. Beyond drug delivery, nanotechnology applications also include photothermal therapy and improved imaging techniques, both of which are crucial for effective cancer treatment. Additionally, it enhances cancer diagnosis by improving imaging precision in MRI and CT scans, leading to earlier and more accurate detection of tumors. Clinical trials and regulatory approvals, including those from the FDA, underscore the growing importance of nanotechnology in transforming cancer care and treatment options. The global market for nanotechnology in cancer therapy is projected to grow significantly, driven by the increasing prevalence of cancer and the demand for effective treatments. Patients and practitioners are encouraged to stay informed about advancements in this field and consider participation in clinical trials for innovative treatment options. However, challenges such as regulatory hurdles and high production costs could limit the accessibility and implementation of nanotechnology in cancer treatment. Despite these challenges, the field of nanotechnology represents a significant step forward in medical treatments, emphasizing how microscopic innovations can improve patient outcomes. Innovative smart nanoparticles are also being developed to release their therapeutic payload only under specific biological conditions, allowing for more personalized treatment. A new study has emerged that could change the landscape of heart disease risk assessment. Researchers from Chalmers University of Technology and Harvard University have found that measuring ApoB-containing lipoprotein particles provides a more accurate prediction of heart disease risk compared to traditional cholesterol tests. This extensive research analyzed over 200,000 blood samples from individuals without a history of heart disease. The findings suggest that standard cholesterol tests may underestimate heart disease risk in approximately 8% of patients. A concerning statistic given that 20 to 40% of first-time cardiovascular disease cases can be fatal. In the UK, current protocols for assessing heart disease risk typically include evaluations of blood pressure, family history, and cholesterol levels. However, these methods may overlook some patients who are at risk. Traditional cholesterol tests primarily measure total cholesterol levels, failing to account for the harmful effects of lipoprotein carriers that transport cholesterol in the bloodstream. Cardiovascular diseases remain the leading cause of death worldwide, highlighting the urgent need for improved early detection and prevention strategies. Fortunately, blood tests for ApoB and lipoprotein A are commercially available, affordable, and easy to implement, representing a significant advancement in heart health screening. Lipoprotein A, a genetically inherited lipid, is another critical marker that significantly elevates heart disease risk and should be included in risk assessments. While cholesterol is essential for healthy cells, elevated levels in the blood can increase the risk of heart disease and stroke, making accurate testing vital. ApoB has been identified as a superior marker for heart disease risk, particularly since standard cholesterol measures can underestimate risk in 1 out of every 12 patients. Early detection of cardiovascular disease is crucial, as many cases can be prevented through lifestyle changes. Jakob Morze, the lead author of the study, emphasizes the importance of understanding different lipoprotein types in assessing heart disease risk, marking this research as the largest of its kind. On April 27, 2025, a significant announcement was made during the American Association for Cancer Research annual meeting regarding a new anti-cancer therapy, ABP-102/CT-P72. This therapy, developed through a strategic partnership between Apro Holdings Inc. and Celtrion, is a tetravalent bispecific HER2 x CD3 T-cell engager. Its design allows it to specifically target HER2 overexpressing tumors while minimizing toxicity to normal tissues. HER2-positive cancers, which include breast, gastric, pancreatic, and colorectal cancers, account for up to 30% of various cancer cases. The collaboration aims to develop and commercialize ABP-102/CT-P72 globally, focusing on these prevalent cancer types. Preclinical in vivo studies have shown that ABP-102/CT-P72 achieved up to a twofold increase in tumor growth inhibition compared to runemotamab, a benchmark HER2-XCD3 bispecific antibody. Dose escalation studies in cynomolgus monkeys indicated that ABP-102/CT-P72 was well tolerated, even at doses significantly exceeding the maximum tolerated dose of its parental antibody. This suggests a broad therapeutic window for the treatment. The therapy is engineered to reduce cytokine release, a common source of toxicity in other T-cell engagers, while still maintaining high efficacy against HER2 high tumors. The preclinical results suggest that ABP-102/CT-P72 may provide superior tumor selectivity, efficacy, and a better safety profile compared to existing HER2-targeted therapies. Data presented at the AACR meeting demonstrated significant tumor selectivity and potent cytotoxicity in models of breast and gastric cancer. Clinical trials for ABP-102/CT-P72 are set to commence in the first half of 2026, with the goal of redefining treatment options for patients with HER2-positive cancers. Celltrion is committed to accelerating the development of multivalent antibody therapies as it transitions into original drug development. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.